Okay, hello, ladies and gentlemen. Welcome to this session of the Alumni Festival 2020. I'm Alexander Bird. I'm well, half of me is currently Director of Research in the Faculty of Philosophy. And half of me is still the Peter Sowerby Professor of Philosophy and Medicine at King's College London. But in just over a week's time, I will be 100% the new Bertrand Russell Professor of Philosophy here at Cambridge. And it's a real pleasure for me to start my new job by talking to you, to the alumni of the university. Okay, so I'm now going to get onto my talk, which is, here we go, called Romanticism, Creativity and the Replication Crisis. This brings together a number of my interests. So I'm a philosopher of science with a particular interest in medicine and medical statistics. I'm interested in the relationship between creativity and rationality in science, and I'm engaged in a project on creativity with my colleague Alison Hills at St. John's College, Oxford. And so this talk brings a number of these things together and shows how they are connected. Okay. So what is creativity? Well, that's contested, or at least I think it should be contested. So there's a standard conception used by psychologists and indeed philosophers interested in creativity, which says there are two things that creativity is about. It's an ability to produce works or ideas that are new, original, and novel, and which also have value. In some sense, you value to somebody or to something or to some goal. So according to two interesting things about this conception of creativity. First of all, it says nothing about the imagination. Imagination isn't essential to creativity, it's just a means of producing novelty. That's been the standard view, but Alison Hills and I think that's a mistake. And so this is what we think is the right way to think about what creativity is. It's a capacity to produce works that indeed are novel, so we agree with the standard view there. But our principal innovation is to say, look, creativity essentially uses the imagination. And importantly, we say nothing about value. And we have a couple of other conditions. Creativity does involve certain kind of fertility. One can't just be creative just once. And there needs to be some motivation to bring one's artistic or scientific product to some kind of fruition or completion. So I say, in this view, creativity and imagination are essentially linked together, but there is no essential link between creativity and producing value. So in our view, it's perfectly right perfectly possible at least, for someone to be creative, but to be producing things that have in fact no value in any sense at all, or indeed might have negative value. One might be creative torture, devising horrible and negative new things. Or one might be an incompetent scientist coming up with hypotheses that are always false and fruitless. Okay, so here's a question. When is it that the creative imagination does succeed in producing works of value? Since on our view, creativity doesn't necessarily produce value, it makes sense to ask, when does it? Under what conditions can it do so? And indeed, what is the value of the creative imagination at all? And want to contrast what we think with what one might call a romantic view of the answers to these questions. So the romantic view thinks that the creative imagination is always good. I mean, one reason the Romantics had is that there's a value, intrinsic value on exercising one's imagination. But here's an instrumental view of why it's always good, because more ideas is good, and more novelty is also good. Just abundance of original ideas is a positive thing. You let 1,000 flowers bloom. Okay, and so the creative imagination is valuable then because it allows us to produce new works. And then on this view, our judgment can filter these new works or exercise itself on these new works, whether new artistic works or new ideas in science, to decide which of them really do have high levels of artistic or scientific value. Okay, so I'm going to talk a little bit about Romanticism, and as we just defined it, as found in science. So science was looked down on by some Romantics, most notably William Blake, but even before him, Immanuel Kant, at the end of the Enlightenment, at the beginning of the Romantic period, thought that there could be no scientific geniuses, and that's because science is a matter of applying rules of reason and logic, whereas artistic genius is a matter of something quite different. It's a matter of imagination and not of logic and reason. However, there were some positive views about science amongst the Romantics. Keats and Coleridge and others had positive view of science, and they wanted to find a place for as it was, a Romanticism within science. And one place we find that is in the work of John Herschel, Cambridge man, indeed a Jonian, I'm pleased to say, and his work, A Preliminary Discourse on the Study of Natural Philosophy. So this is John Herschel as a young man looking suitably romantic, and here is him later in age, looking every bit the romantic mad scientist. In a rather wonderful photograph by Julia Margaret Cameron, the great pioneer of photography. Anyway, so Herschel's idea was that there's a place for the imaginative in science, and that place is in the process of coming up with new scientific ideas, hypotheses, theories. So he made a distinction that philosophers of science subsequently have called the distinction between the context of discovery and the context of justification. Now, these aren't desperately well named, good terms for the distinction, but here's the idea. So the context of discovery is the process of coming up with a new hypothesis. It's just a matter of exercising the imagination to think of new explanations, new ideas, new theories. And this contrasts with the context of justification, which is the matter of deciding whether any particular hypothesis is well supported by the evidence or is refuted by the evidence, was shown to be true by the evidence. That's the context of justification, and that is the study, and understanding that is the subject matter of philosophy or logic broadly conceived. Whereas the processes that go on in the context of discovery, the thinking up of the new ideas in the first place, that's got nothing to do with philosophy. That's the subject matter of the psychology of science. So here we can see that we can get the romantic into science, because the scientist is every bit as imaginative as the artist in the first of these processes, the context of discovery side of thing. But the bit that Kant thought was distinctive of science, the logic part, so to speak, that's in the context of context of justification, and that's the creative imagination, according to this view, doesn't play a role here at all. I happen to think that's a mistake, but I won't dwell on that in this talk. So we've got this nice this distinction, and so you might think, look, here's the romantic idea again. We should encourage the creative imagination in the first of these to think up lots of new hypotheses, many of which are very original, and then we can use logical methods to filter out the false hypotheses, leaving just the true ones standing. Indeed, that's a view, and I'll come to mention this a bit later, that we associate very much with Sir Karl Popper. So I regard him as a romantic in the philosophy of science. You have lots of bold hypotheses, and then we try and falsify them, and that's in a sense sort of focuses our minds on those that are have some chance of being true. Okay, so this is Romanticism as applied to the philosophy of science, and I want to talk a little bit now about why I think that that's a big mistake. Okay, so why more creative imagination isn't always better in science. And you'd have thought that if this picture on this slide is right, then the more you can get into the more hypotheses you have, more original they are, the better, because the filtering out in the second part, in the context of justification, will end up telling us which are true or least likely to be true. But I think that that's a mistake, and we don't always want more creative imagination. So I'm a bit of an anti-romantic in this context, at least. Okay, so what I'm going to do is to explain why I think, as it were, too much imagination is responsible for something called the replication crisis, also known as the reproducibility crisis. Okay, so what is this crisis? For those that don't know what it is, so here's the problem. In certain fields in science, there are published results, often well-known, admired published results, and people then come along later and try to replicate the original experiments, or ones much like them, and then they get very different results. Where the original studies found some large positive effect, the new studies find no effect at all, or they find an effect, but it's a much smaller size than the original. Now, of course, science isn't perfect. One might expect this to happen from time to time, but the replication crisis arises because it happens really far too frequently, and in particular in clinical medicine, an area that I have particular interest in, but also in psychology, most especially social psychology. So in these areas, one can find studies, replication studies, or meta-analyses of replications, which find that a third, or sometimes even a half, of original studies, published studies, have been replicated without success, without finding the original results. So it looks as if a large proportion of these published results are at best unreliable and quite possibly simply mistaken. Okay. So that's what the replication crisis is. And I want to get into explaining how it arises. Now, one thing that makes a bit of a crisis is why people think it, other reasons why people think it arises. And the most popular explanations for it tend to point to as a moral crisis in science. This all happens because scientists are engaged in what are known as questionable research practices. It basically sort of cutting corners in various ways, or doing things that one ought not, like p-hacking, which is generating lots of data and then trawling through it to find a correlation that looks statistically significant, and then publishing that, and various other things that we ought to frown on in science. I'm going to argue that that isn't the only possible explanation, and indeed I think that it's important to recognize that even if all scientists were perfect saints and didn't engage in these practices, we would still have much of the replication crisis left. So it's not just a matter of sort of bad scientific ethics, but it's also a problem of, well, as I explained, too many bad hypotheses being tested, as it were, being over-imaginative, creating new hypotheses. And if a lot of them start off, turn out to be false, then we're going to get this kind of failure of replicability in any case, however honourable and proper the scientists are. And the way I'm going to explain that is by an analogy. It's a perfect, statistically speaking, it's a perfect analogy for something else which is a little bit of sort of puzzle in medical statistics. It's only a small puzzle, but it's it goes like this. It's something called the base rate fallacy, and it's sort of understood by asking yourself this question. You imagine that you go for some kind of medical test. It could be a screening program for prostate cancer or something like that, or breast cancer, whatever it might be. Or in my example, to date, a test for whether you've got COVID-19 antibodies. So what we're going to imagine that this test is pretty reliable, and you test positive, and then the question is, do you have the antibodies? So I've imagined that this test is one, we're carrying it out when about 1 in 10 of the population have these antibodies. This test has what we call a specificity of 95%. Now, don't spend too much time on all these statistical terms. What that means is does this test give a positive response just to these antibodies, or might it give a positive response to something else as well? And this test has a specificity of 95%, which means it responds really only to the antibodies, and it responds to something else in only 5% of cases. So that's to say, if somebody take 100 cases of people, none of whom have the antibodies, it will give us the correct negative result in 95% of cases, and give us a positive result, false positive result, in only 5% of cases. And it has a sensitivity of 80%, which means to say that of all the people who do have it, you say 100 people who do have these antibodies, it will correctly tell us that they've got the antibodies in 80% of cases, and give us false negatives in 20% of cases. So we can see it's a pretty good test in both of these respects. So you test positive, and so what's the probability that you do have the antibodies? That just shows that you've had COVID, and perhaps you've any luck are immune from now on. Well, you might think, well, look, it's a good test, 80%, 95%, somewhere in between the two. What's the right answer? Well, actually, it turns out to be something rather different from all of these. So what we're going to do is imagine 100 test subjects chosen at random from the population. Now, I said that 10% of the population have these antibodies, so they're represented by the red dots there. And the black dots are 90 of them. Those represent the 90 people without antibodies. Okay, so we give everybody our antibody test to see whether they've got the antibodies. So let's think first of all about the 10 subjects with the antibodies. I said our test was 80% sensitive, which means it will pick up on 80% of these and give them the correct positive result, and it'll miss out on 2. So we get 8 positive results from these 10 subjects. Of course, there are 90 subjects without these antibodies, and I said our test was 95% specific, had a 5% false positive rate. So 5% of these will give us a positive result, even though they don't have the antibodies. Okay, so that's about 4.5. Okay, so we've got these 4 positives and the 8 other positives. So what we've got overall are 12 positive results, 8 of which were the correct true positives, people who've got the antibodies, but 4 of whom were the people without, the 5% of the people without the antibodies who gave a false positive. So we've got 12 positives, 4 false, 8 true. Okay, so now let's go back to our question. You get a positive test result. Does that mean you've got the antibodies? Well, as you see, there's 8 out of 12 chance that you've got the antibodies, and a 4 out of 12 chance that you don't. That's to say, you've got a 1/3 chance that your positive result is a false positive. Okay. So yeah. So although this test was a really good one, 95% specific and 80% sensitive, nonetheless, it's the case that in this context, you've got a positive result. There's a 1/3 chance that you don't, in fact, have these antibodies, and that's sort of bad news, because you might go around thinking, look, I've got these antibodies. That shows I can't get infected, and actually, there's a 1/3 chance that you are entirely susceptible to COVID. So this can be misleading, and the reason is that if we concentrated just on the reliability of the test and thought, a reliable test, therefore it's highly likely I've got the antibodies, what we've ignored is the effect of the background rate in which antibodies are found in the population. It's because actually a large proportion of the population don't have the antibodies that we get a modest but not insignificant number of false positives, the black dots, the 4 out of 12 black dots. Okay, so that's what's generating this little puzzle. The fact that there are a lot of people without the antibodies, some of whom will show up as false positives. So now I'm going to talk about science, about testing scientific hypotheses. So we're moving from testing people for a disease or these antibodies to talking about testing theories and hypotheses or whether they are true or not. And I'm going to draw a parallel between these 2 cases, and the statistics, that outcome we saw will show up here exactly the same way. So how do we test for truth? Well, a standard way of doing this in psychology and in clinical medicine is to run a randomized controlled trial. 2 groups, one who has so you're testing a new drug to see where it works. You give a control group with a placebo, experimental group is given the new drug, and you see whether there's a difference in mortality or difference in how long it takes for individuals to recover and be discharged from hospital. And you see that there is a difference, that there are fewer deaths and quicker recovery in the experimental group. But we know that these things can come about by chance. So we have to do some statistics just to work out whether we regard the difference between these two groups, experimental group and control group, whether we regard that difference as statistically significant. And we have a statistical method for doing that, and it's called null hypothesis significance testing. And the standard way of doing this is a randomized controlled trial with what we call a significance level, or alpha, of 5% and a power of 80%. And we can regard this as a test for truth that has a specificity of 95% and a sensitivity of 80%. So that's our standard mechanism in these fields. And so it's not a surprise I chose 95 and 80% for my previous example. That was there on purpose. And there are some reasons, although this is a bit of a guess, but an educated guess or estimate, that about 10% of hypotheses that are tested in these scientific fields are true. I mean, it's going to vary from area to area, but yeah, one in 10 new ideas that you reach the stage of a randomized controlled trial that it might produce a publishable result. We reckon that 1 in 10 of these is going to be a true hypothesis, and 9 out of 10 is going to be false. Okay, so we've got the same. This is the picture we had with our 100 test subjects with antibodies, or 90 without antibodies and 10 with. But now we're going to imagine that we're not test subjects with or without antibodies, but they're hypotheses, that some of which are true and some of which are false. And as I said, we estimate about 10% of these new hypotheses coming up for testing are true, and 90% are false. But it all works out in the same way. We get 12 positive results, 4 of which are false and 8 of which are true. So what that means is that this null hypothesis significance testing that we do with a randomized controlled trial that will produce a positive result that in fact turns out to be a false positive in 1/3 of cases. But you've got a positive result, okay? That's, yeah. You didn't know it's false. You think it's likely to be true. You get a positive result. It's publishable. You send it off to the Lancet. It seems to publish anything these days, or whatever the journal is in your field. You get published. People read it and think, oh look, these scientists have shown X, or it's very probable that X. They've done an RCT with null hypothesis significance testing in exactly the way that scientists expect that to be done. They get a positive result. That's indicative, strongly indicative of that this effect exists, that this medicine works, or this effect in psychology really is there. But in fact, 1/3 of those cases, there'll be no effect. The medicine doesn't work. Or if there is no effect in the psychological case, it's just an artifact of the statistics. And the fact that of the hypotheses being tested, 90% are false. And so we'll get a small proportion, but not insignificant, of false positives, these 4 false positives out of the 12 positives that there are. So if that's the case, then it's not surprising that when people come and try to replicate your original result, or these original results, in the 1/3 of cases where the hypothesis is in fact false, it's hardly surprising that the attempt at replication doesn't come up with the supposed effect that they found in the first place. So the conclusion of this bit of my talk about the replication crisis says, look, at least some of the widespread failure to reproduce positive results arises because randomized controlled trials with null hypothesis significance testing is going to produce non-negligible, a sizable proportion of false positives. If, this is the point, generating a true hypothesis is rare, it's because 90% of the hypotheses we're testing are false that this effect arises. It wouldn't arise if half of them were false and half true, or even smaller proportion were false. And so the conclusion that I draw for our work on creativity is that more novelty, more creativity, more imaginative hypotheses, both more in terms of numbers, but also more in terms of more radically new, that's going to undermine the reliability of science. 1/3 or thereabouts of published results will turn out to be mistaken. And so one conclusion one might want to draw from this is that perhaps imagination needs to be constrained a bit in order to produce value, where value means true outcome. Perhaps we don't want to have quite so many radically new hypotheses. If we had fewer, then the positive results will be more likely to genuinely indicate truth. Okay, so that's my explanation of why I think that it's not always the case that more imagination is better in science. But what about the arts? Surely the arts are different. Imagination in the arts, it's always a good thing, surely. Well, Alison and I think that something similar might be going on in the case of the arts as well. So here's our sort of hypothesis that we're working with. That as your works of art become easier to produce and to experience, the quality of even the most successful work will tend to decline. Why is that? One reason is that the more work that there is that is produced, the less time there is for us to be able to engage our critical faculties with any one piece of work. The time available for engaging with each is going to be less. Therefore, the reliability of our judgment is going to decrease, and correspondingly, artists are going to be motivated to aim for easily accessible attractiveness over the kind of more profound value that requires extended engagement to be properly appreciated. So what's the evidence for this? Well, there's quite a lot of sort of anecdotal evidence or qualitative evidence. You're talking to both to artists and to critics, that they find that the overwhelming quantity of work that's being produced actually does indeed make it more difficult for them to engage properly with any of it. So slightly more quantitative evidence is available, for example, in this paper by Sarah and colleagues, who looked at the more successful pop songs, those that get into the top of the charts, over a 55-year period. And they showed that these songs have, over this period, shown a reduction in these three musical qualities: timbral variety, harmonic richness, and dynamic range. I mean, to put it bluntly, their evidence is that pop songs have become more boring over time, more similar to one another, showing less of the features of music that tend to make it interesting and tend to be found associated with quality. So there's some quantitative evidence as well. Okay, well you might think, well, okay, that's true for sort of run-of-the-mill work in the arts, but it's not going to apply to the exceptional work, the work produced by geniuses. Kant and the Romantics that followed him put a great store by artistic genius and felt that they were exceptions to many of the rules that govern the rest of us. So, for example, there's this famous letter by Mozart that seems to exemplify this. So, Mozart tells us that that on occasions, certain occasions, his ideas flow abundantly. Whence and how they come, I know not, nor can I force them. He says, all this fires my soul, and provided I'm not disturbed, he is able to produce the whole of a work, though it be long. It stands almost finished and complete in my mind, so that I can survey it like a fine picture or a beautiful statue at a glance. I don't hear in my imagination the parts successively, but I hear them, as it were, all at once. When I proceed to write down my ideas, I take out of the bag of my memory what has previously been put into it. For this reason, the committing to paper is done quickly. Everything is already finished. It rarely differs on paper from what it was in my imagination. So if you're a genius like Mozart, then you're going to produce works in this way, almost God-given. And they're not going to be affected by any of the things I've been talking about. Well, it's interesting. So this letter by Mozart, well, turns out to be a forgery. It was published in 1815, the height of the Romantic movement, really, but later found to be a forgery. Despite being shown conclusively to be a forgery, many people continue to cite this letter as an example of what it is for a creative genius to work, how it's different for them from what it is for the rest of us. And indeed, some philosophers have been amongst those who continue to cite this forged letter. I mean, the truth is, Mozart sometimes found it difficult to compose, and he struggled in the way that the rest of us do. I mean, the outcome was better, but but not always. And so I think this idea of exceptional genius is a romantic idea. Is that what's generated that letter's Romanticism? But it's just simply false. Okay, so just to conclude, some positive thoughts. So on the view that I've been putting forward, perhaps it's good to constrain the creative imagination, not let it run riot in the romantic way. Well, what should do that? Well, I appeal to the work of Thomas Kuhn, who identified what he called an essential tension in science between the importance of building on tradition, yet of course science needs to innovate. And he found a tension between these two. Too much emphasis on tradition, we don't innovate enough, but we overly innovate, then the chances of producing something that's scientifically worthwhile are reduced. And so that's a view to which I'm very sympathetic. It's quite a conservative view in a way, and it stands in contrast to the work of Popper, which I briefly mentioned earlier on. Popper wants scientists to generate bold hypotheses, many bold hypotheses, and we use the method of falsification to eradicate the false ones. So in a sense, oddly enough, although Kuhn famous book, Structure of Scientific Revolutions, and associated with the term revolutions. Actually, Popper, who's much more revolutionary, I mean, he is the Trotskyite of the philosophy of science, because he believes in sort of permanent revolution, new bold hypotheses all the time, and then we cut them down with falsification. So the hypothesis that Alison Hills and I are sort of investigating is this, that even central innovations, revolutionary advances in the arts and sciences, actually often draw on insights already present in the tradition in some form or other. And we work through various examples in science, relativity and quantum mechanics, revolutionary yet build on things that are already actually present. Cubism, I think, is true. Painting, you can do that. So I'll just briefly mention the new Viennese school. So this is the sort of the serial technique, 12-tone technique in musical composition. It throws out tonal composition, very radical, but one can see really what has happened as a development really of the increasing chromaticism found in 19th century music. So in the classical era, chromaticism is just that, literally a little bit of coloring these chromatic notes. It grows and grows through the 19th century until the new Viennese school, Schoenberg, Webern, realized that what was had been an incidental feature can be made the basis of a new musical language. So although it was revolutionary, it nonetheless built on something already found in the tradition in which they were working. So one of the ideas we're trying to develop is this idea of the way that tradition can be when it can be a useful constraint on the imagination, in order that we can have a higher chance that our new ideas will have value, whether that's in science or in the arts. So that's the end of my talk. Thank you very much for listening. And what I'll do now is I will stop sharing my screen and look at some questions. Lots of really good questions here. Okay, one question was about this base rate fallacy I was talking about. It says, in order to know the base rate, we need to test people first. And in order to know the probability with which a given positive result is a true positive, we need to know the base rate. Correct, that's right. So how do we get out of this vicious circle? Just by multiple testing of the same person? Well, yes, exactly right. Yeah, I mean, that is the best way to really check that you've got your base rate correct, is to do repeat testing. That is definitely the best way of doing that, and iterating that can get one closer to giving one a correct estimate of what the base rate is. But then Alison asks, how accurate is my estimation of the base rate in science, that 90% of hypotheses are likely to be false? So what I just referred to, this sort of multiple testing, is less easy to do in science than it would be for something like a test for a disease. So I'm not entirely confident that this 90% figure is right. We can make some inferences from these replication studies, and we can get evidence from what goes on in laboratories. There's anecdotal evidence of what scientists think. So, I mean, it could be 95%, could be 80%, and I suspect it will vary very much from one field to another. And I think that some sciences, that actually, so in high-end theoretical physics, the hypotheses put forward for testing are much more likely to be true, because they're, for various reasons, the filtering processes work at an earlier stage, because you don't want to build a large hadron collider at multiple billions of pounds to test the hypothesis. that's false, but also because there's better grounding in solid theory, well-established theory. Okay, so yeah, that's a good question. Well, they're all good questions, in fact. Publication bias. Right, good. Anna has asked, would publication bias play a role in the replication crisis? Right, so many scientific findings which don't find statistical significance against the null hypothesis, there's nothing here, okay, are rejected, and this can contribute to the lack of reproducibility. I think that's right, but only up to a point. So publication bias is a real problem. So the point is, so you've done your experiment, your RCT, and you don't get a significant result. Journal's not interested in this boring negative result, so it doesn't get published. Or it's not going to do your career much good. You move on to something else. You put it in the file drawer, as they say. How much does that contribute to this problem? Well, it doesn't directly contribute, because what's going on there is a failure to publish a true negative, whereas the problem of replication is the existence of false positives. So it isn't a direct effect, but it does contribute, as Anna says, to the lack of reproducibility, because if there are some negative results regarding a particular effect or hypothesis out there, but not known, then we'll go in and do our one. We might just replicate that and then get by chance this positive result and publish that. Whereas if the negative results had been known in advance, then we might have been more likely to think that our positive result might be false positive. If the negative positive are published, then my positive result can be put into a meta-analysis, systematic review, along with the negative results, and will overall get the right answer. You'll get towards the answer that in fact there might not be much of an effect at all. But if only the positive results are published, then we'll get a skewed view of what the truth is. So I think publication bias is a problem. It contributes to the lack of reproducibility, but isn't directly its principal cause. Okay, so Richard says, good scientists and artists are not being creative at random. They are using their judgment in the light of their extensive experience. Doesn't extensive experience channel their judgment in more promising directions? So, for example, Darwin's experience of of human breeding of animals led him towards the idea of natural selection. Yeah, I think that's exactly right. Yeah, so I mean, in the sense that links in exactly with what I was saying towards the end about, as it were, the importance of working within a tradition. Yeah, in some ways, I was talking about, as it were, the extensive experience of not just oneself, but the tradition one's working in. And Richard's question is emphasizing as the experience of the individual scientist. And I think that both of these are important. So you're absolutely right that. So take that example of Darwin. So he used artificial selection, as the model for natural selection. So it's interesting that the very first chapter of The Origin of Species isn't about the natural world at all, or about natural selection. It's about artificial selection, but it gives us a model by which we understand and can see the way that natural selection works. But I think that's exactly what we're trying to say, that we work, we are directed towards true or productive ideas by making connections with what has worked in the past. And I think that's to contrast with at least one version of the romantic view, which is what one wants to do is to empty one's mind of precedents, of traditions, of exemplars from the past. This working with our past experience, that's just too conservative. And one wants to, they had various ways of thinking about this. You try and think like a child. You've not got experience. That was one image that the Romantics had for how one should be more imaginative, to think like a child. Of course, others took drugs as a way of freeing themselves from what was already in their mind. So I entirely agree with Richard on this point. Okay, and there's an anonymous question. Why do I think that medicine and social psychology are more vulnerable to incorrect hypothesis generation than other branches of science? Okay, so I think that the reasons are different for medicine and for social psychology. The reason for medicine is it's bloody complicated. The human body is fantastically complicated, and despite all the research we've gone into it, really understanding quite how things work, all the feedback mechanisms and so forth, is actually really quite tricky. And so what the primary research, just basic research into physiology and pathology, gives us is an incomplete picture of the bodily mechanisms, or the mechanisms of disease. So that incomplete picture is able to generate hypotheses about what might work, sensible thoughts about, try this, it could work. And in my published work, I've got examples of how drug development for treating Alzheimer's has worked in this way. Sensible ideas, but it turns out the drug just doesn't work, and you can find out why after the effect. It was a good idea, but not one that really stood a very high chance of success given our incomplete knowledge of the real details about how human physiology works and is sensitive to certain kinds of change and feedback effects and so forth. So that case is that although we have a lot of background theory, it's incomplete. Social psychology, I think, is a bit different. There, I just think that the field is just too young. There's no underlying theory at all there, really, or very little underlying theory. And your hypotheses just come from, really come from nowhere. Often they come from, a bit like the modeling I was talking about earlier. They are modeled on what seem to be successful hypotheses elsewhere in social psychology. But part of the problem there is that if the earlier ones turn out not to be right, but they're victim of what we've been talking about already, then as it were, one could be modeling new ideas on apparently true, but in fact false ideas. So your very famous effect, alleged effect, this power posing idea that you can, if you adopt a powerful pose, that increases self-confidence, it increases various biomarkers for positive effect, testosterone things, I think. Anyway, so that was very famous. I mean, one of the biggest YouTube sensations was a presentation based on this research. That was wasn't grounded in some well-established theory. It was modeled on another idea that if you grab a pencil and smile in a way that exercises one's smile muscles, then in fact that gives you more positive thoughts. So there's an earlier study that gave that result, that you might think positive thoughts cause you to smile, but actually it turns also that you can reverse that effect, that by smiling, exercising the smile muscles, you can have some more positive feelings. You could see the analogy between the two, but it turns out actually that the smile effect actually can't be reproduced either. So yeah, that's the way. This all contrasts, say, as I briefly mentioned, with physics, where you say, for example, you take the Large Hadron Collider and you're looking for the Higgs boson. Look, there, the Higgs boson hypothesis was one that pretty well all the physicists thought it was true, and that this experiment would find it. Why is that? Well, that's because the Higgs hypothesis is actually a piece, in fact, one of the last pieces of a big puzzle, which is known as the standard model of particle physics. And this model has been around for some decades. It has been massively well confirmed by all sorts of experiments working on different bits of this model. And the Higgs boson is one very important part of this picture. I mean, non-trivial exercise to develop the idea, but it fits very neatly into this pre-existing grand theory, which is super duper well confirmed. So in the light of that, everybody expected the Higgs hypothesis to be correct and to be proven to be correct. So physics, I mean, it's not saying physics is great. It's just, I'm found medicine, but it's just that much more difficult in those fields, in medicine and psychology, to generate hypotheses that have a chance of being true than in physics. Okay, look, it's 1:29, so I have one more minute left. I don't think I'm going to be answering any of these more very interesting questions. Oh, it's such a shame. We could be here for another hour answering fantastic questions. Look, so I'm going to say thank you very much indeed for listening to me. And as I say, all these questions do show me that that I got across some of my ideas. I hope you enjoyed that. Thank you very much for attending. I should tell you that there are still other sessions that you can book for. So if you've enjoyed this one, please go and find some others to enjoy as well. I hope you've got something out of this. I think so from the questions. And have a very good rest of the day, and enjoy the other Alumni Festival events to which you will be going. Thank you very much for attending.